So I'd like to welcome Dr. Naupaka Zimmerman. Naupaka Zimmerman is currently an associate professor in the biology department at the University of San Francisco. He is interested in community and ecosystem ecology with a primary focus on plant-associated microbes in natural and agricultural systems. His PhD is in biology from Stanford, and his undergraduate degree is in environmental science and public policy and anthropology from Harvard. In addition to ecology research, he has long been interested in computational pedagogy and reproducibility, and has been volunteering with software and data carpentry for over a decade. This fall, he will be starting a new position on the faculty of the Environmental Studies program at the University of Kansas. And I also want to make a personal note that I told Naupaka earlier that the reason I am able to do R is because of him. I went to a workshop that he held at the Ecological Society of America a number of years ago, and it was so crystal clear in terms of how to work with our code and the different kinds of things you could do that it ended up being the kind of turning point for me in finally shifting away from SPSS. So I really appreciate that. So we can welcome Naupaka. So let's see. You'll need to share.
Live share screen? Yes. So you can go ahead. Well, thank you for the very kind introduction. Particularly during my postdoc, I really got into these teaching R workshop things, and I was doing like 4 or 5 every USA. It was just went a little bit to the deep end, but they were a lot of fun, and it's good to hear. This is called icing. Yes. And that's great. Very good. Okay, hello everybody. I'm Naupaka Zimmerman. I'm technically in 2 jobs at the moment. I left San Francisco in my car less than a week ago, moving to my new position in Kansas. I've been in Lawrence, Kansas now for less than a week. But very happy to be here with you all today. And I struggled with the title here a little bit, but basically this is like things about macro system science that make me excited, and also about teaching. Like this is very much of like, what about macro systems gets me jazzed? Before I sort of dive into this, I sort of want to preface it with a good amount of this, not all of it, but a good amount of it is part of this grant that's just finishing up with a colleague of mine, John Vilbitz, who is on the faculty at Augsburg University in Minneapolis, and this is a project that we're just finishing up. So I know many of you have read this paper. I saw that it was something that folks are asked to read, and that maybe it has been discussed before. But you know, in my years interacting both with the macro systems program at NSF and with colleagues and people at ESA meetings and that sort of thing, it's always discussion. It's like, you know, what is macro systems? Like, what makes it macro system science? And I think this paper is nice because it kind of wrestles with a lot of those ideas. And in my mind, it has to do with both scale and type. So it's thinking about larger scales over space and over time, and also these connections across these sort of hierarchical scales. Um, so when you think about something like this, the example from the paper was thinking about clearing of the Amazon rainforest and all the things that intersect there, the ecological dynamics to lead to land clearing in the Amazon. A little bit closer to home for me, so this is a picture from the Sierra. This is just outside of the northeastern boundary of Yosemite National Park near Tioga Pass, for those who are familiar. And this is a sort of natural mountain system, right? And this is the sort of thing that I think many classically trained ecologists, this is like the amazing summer research that you go off to do. And we have faculty in my, one of the faculty in my department at USF who goes up every summer and studies Belding's ground squirrels and behavioral ecology in this area of the park. And has done that for 30 years and is very interested in these dynamics of these ground squirrels. If you're a vegetation ecologist, you might study dynamics of tree recruitment or competition, or if you're an aquatic ecologist, there are all of these lakes. There's all these sort of really interesting ecological things going on that you could look at this scale, and that might be, if you're sort of zoomed out here, this might be landscape ecology, right? If you zoom in a little bit, now you might be thinking about ecosystems or communities, these sort of smaller scales. But sort of on the way to the Sierra, if you're driving from San Francisco anyway, you drive to the Central Valley of California, right? And so this is a picture of almond trees, which cover a whole lot of the Central Valley of California, and are exceedingly water use intensive. They need a ton of water irrigation, and if they have a ton of water, they grow very well and always make good money for the good folks in the Central Valley. The problem is California doesn't necessarily always have a lot of water, and that water that it does have comes from places like this. So if you're up there and you're an ecologist and you're thinking about what's going on in the dynamics here, and let's say maybe you're studying some sort of insect pathogen outbreak that's causing tree dieback. This also happens in the Rockies. That does things like change the albedo, and it changes the patterns of snowmelt, and it changes when all that water funnels down into the Central Valley, which changes how angry the farmers are when they have to try and get the water from the state, which is withholding it because they're trying to throttle it so it lasts through the growing season, right? And this is where I start thinking that we're getting into macrosystems ecology, right? You're thinking about these things at these larger scales. You're thinking about how these sort of local ecological dynamics really start intersecting in some really interesting ways. So that's sort of on one side of my recent travels. And here's another one. This is a picture from Quonset Prairie, which is in the Flint Hills in sort of central eastern Kansas. It's sort of spectacular remnant native tallgrass prairie. And the reason that it is native remnant tallgrass prairie and that there's only remnants of it left is because the soil subsurface is exceedingly rocky, and so it can't be plowed. And so the rest of the Midwest, which was plowed very vigorously, and all of the amazing deep prairie-rooted native grasses that had evolved there for millennia were all ripped up and replaced. But in these places where the geology is just so, and you couldn't plow, now you have these remnant patches, and instead of having their native herbivores, they're now intensively grazed by cattle. But they serve a very similar ecological purpose. And so you start getting these dynamics where you have intersections across many different states, across regions, where you're thinking about the social, where you're thinking about the ecological, where you're thinking about the geological, and that starts getting in that microsystem space. So the next thing that I think I wanted to sort of introduce to you is this talk is organized a little bit where I want to tell you kind of where things are going next, and then I want to walk backwards and tell you how we got there. Is sort of how we're going. So understanding a macrosystems scale thing like agriculture in the Central Valley of California and its ties to the Sierra understanding something like agriculture across the entire Great Plains, something like that is a challenge in and of itself. But I think a real challenge for ecology as a discipline, certainly compared to many of the other sciences, is thinking about how do you predict, how do you forecast, how do you go forward? And many people say that being able to do that means you have a pretty good understanding of how things work. And that's been a really hard problem for ecologists, right? Ecology is a very, very complicated discipline, and there's so many things going on. And it's really pretty hard to look forward. If you ask most people, thinking about forecasting and looking forward, um, climate, weather, these sort of things come to mind, and they've actually been remarkably successful. Like, I mean, if you check the weather, it's like really pretty good for 3 or 4 days at least, usually, in most places, right? And you even get like sometimes a couple weeks worth of weather information that's pretty good. The meteorology folks have been really, really good at being able to do this. And in ecology, over the past couple decades, there have been these sort of intermittent, but relatively consistent pushes to try and get ecology moving in that direction, right? So this top paper is from 2001, talking about ecological forecasts. This bottom one is 2018, a little bit more recent. And these are sort of high-profile position pieces, basically saying, really, ecology should try and push in this direction. And I think that when we think about sort of synthesizing in our head like all of these different interacting principles and like putting them into some sort of quantitative model that then provides use back to society, I think that this is a potentially useful framework to do that. And I'm using all of that to sort of set up this much more recent initiative. This is 2023. Some of you may have heard of this. This is the Neon Ecological Forecasting Challenge, run by primarily out of this ECHI group, which I'll discuss in a second, led by Mike Dietz at Boston University. And there are two things here. So some of you are 100% familiar with NEON and 100% familiar with the Ecological Forecasting Initiative, but not everybody, maybe. So there are two parts to this. The first is the Ecological Forecasting Initiative. This is started as at NSF, RCN, and may still be, just like MEFA. But basically, the idea is to take ecological information and use it in a sort of data assimilation framework to try and predict forward a whole bunch of things about ecosystems. And that could be things like beetle population dynamics, or it could be things like soil respiration, which I'm going to talk about later, or it could be any other thing that an ecologist might care about. But it's this express idea of like, how accurate can we be, and with what kind of error bounds, looking to what's next. Not just explaining about what is, but what is going to happen based on these inputs. So that's one piece of this, and the second is NEON. I'm hoping that all of you are familiar with NEON, the National Ecological Observatory Network. I'm a huge fan of NEON, which you will certainly get the sense of by the end of this talk, if you haven't already. But NEON is this phenomenal resource for ecologists in this country, and I think around the world. It's basically this distributed network of sites that all have similar infrastructure and data being collected by people using standardized protocols. And then all of that data is released totally free of charge on a public domain license, CC0, for anybody to do anything they want with, even without a login. It is astounding that they do this. And the data interoperability across data sets is also really impressive if you know how to do it. And we do have an expert in the room, Dr. Claire Lynch, who does a lot of this stuff for NEON. So if you have not interacted with her, I would take advantage of that this meeting. Okay, so how does this all work? So this is an example of a NEON site. This is the Kanza Prairie. And the first thing you want to do if you want to predict something is you've got to measure the way it is right now, right? Like you have to get the state currently before you get the state tomorrow. And so this is an example of NEON site. This is their flux tower in the top left. And the idea with this forecasting initiative, this is a figure from the paper announcing that initiative, for folks who are not familiar with this sort of thing, basically they're taking data from these observatory networks, primarily NEON in this case, but the framework is general. You're getting that data, you're using it to with a set of forecasts, and you're forecasting out that data, which is then evaluated against what actually happens in a week or 2 weeks or whatever. And then that's used to feed back to update the models, right? It's a sort of data assimilation modeling framework. And they have these people that basically they've set this competition up so people can submit their models, their forecast models to this using NEON data. And they're sort of competing against each other and like who's doing the best job trying to predict what's coming next. And so some people might use mechanistic process-based models, other people might use machine learning models, but it's this idea of like how accurately can you forecast and with what kind of error bounds. And this is leading to say that my colleague John Zobitz, I am not a part of this grant, but just a couple of years ago got a grant to sort of build on what I'm going to be describing to you today to use what we've done with soil respiration at NEON sites in this forecasting frame, right? So all of the work that I'm going to be describing to you in my head in the sort of framework that I'm thinking of it is feeding into the ability to now use at scale, both over time and over space, through the strength of the NEON network to predict this thing, soil flux, that people might care about. But this is, again, in my mind, a general way of thinking about the power of macrosystems principles, not only macrosystem science principles, but also macrosystems data principles, which I think NEON has done an excellent job in sort of advancing within the ecological community over the past decade or so. So, okay, so all of that said, that's sort of where the edge of this project is now, but now I'm going to rewind a little bit and sort of tell you the story of what we did to get there. So a little bit of background first. So this is Bio 101, freshman year. Some of you may have flashbacks. But thinking about the carbon cycle in a very simplistic way, right? So you have photosynthesis, carbon coming in, and I admit that most of this presentation is terrestrially biased. That's my expertise, so I'm sorry for you audio people out there. But so you have carbon coming in, you have respiration, carbon going out, you have that going into the soil, and so forth. Pretty simplistic. And if you want to measure that at the scale of an ecosystem, right? Before this idea of a macro system, like ecosystems are kind of the big thing that ecologists are thinking about. The way to do that is generally with an eddy flux system. So many of you, again, may be familiar with this, but basically by monitoring the airflow eddies very, very rapidly and the concentrations of carbon dioxide and other gases that move up and down over and through vegetation canopies, you can get a sense for how much carbon is coming in and out of an ecosystem. And this is very cool. It's a little bit fuzzy, and there's a lot of math involved and a lot of processing and a lot of judgment calls. But it works pretty decently well. And what comes out of it is a lot of really useful stuff, larger scales, right? So you have time-based information for inventory needs. If you're trying to think about forests taking up carbon or sequestering it or losing it because of drought or whatever else. It allows you to quantify disturbance more precisely. It helps with decision making. And again, coming back to this idea of using this these constantly flowing data streams to help us validate these and parameterize models. And they could be global models, they could be global climate models. Oftentimes this type of stuff is used for that. But it could also be more local models, or it could be models at a macrosystem scale, or at an ecosystem scale, or even at a local scale. So if you're breaking that apart, so this is a little bit more about the CO2 fluxes that you might get in a standard terrestrial ecosystem. Basically, what the eddy flux tower is capturing is this balance between the ecosystem respiration going out, right? And then the gross primary production, everything that's getting fixed by photosynthesis, by the autotrophs coming in. And the net of those is this net ecosystem productivity, also sometimes net ecosystem exchange, right? Which is the balance between the ins and outs of these boxes. And so when you think about this in a neon context, to like put this in a real world and not box and arrow diagram, this is the eddy flux tower at Kansa. The canopy is not very high at Kansa. This is the tall grass prairie. It was recently burned. This was in June, so it hadn't gotten to be as tall as it sometimes gets. But these eddy flux towers need to be well above the canopy, and this one is certainly. That's just the tower. But if you go inside of the building here, these are some undergrads giving me the side eye as we talk about all the cool instruments on this field trip. But each of these neon sites are set up exactly like this, right? So all over the entire country, Puerto Rico, Alaska, Hawaii, if you have a neon terrestrial core site, it's set up like this. You will have an eddy flux tower, you'll have this instrument shed, you'll have a whole set of soil plots, which I'm going to talk about in a second. If you have not visited one of these sites, then the people are very friendly, and I would highly encourage it. Whether you go with your class, if that's possible, or you go yourself just to like poke around, like it's really neat if you're a science nerd, and I think we're all science nerds in this room. So inside of the shed, they have all of these reference gases that they use for calibrating the machines. They have all of the data storage and networking. These sheds are generally air conditioned, so if you need a little break during the day. And they're not just measuring concentrations too, right? Like I'm not sure how many folks are aware of this, but they're using cavity ring down spectrometers in all of these sheds. So they're measuring basically in real time continuous isotope measurements from these gases at these different places across the towers. I mean, this is just super cool data. And again, totally free. Anybody can access this. The challenge with the whole eddy flux setup, and this is true for neon, this is also true for FluxNet and basically any other eddy flux type setup, is that it's really hard to differentiate between this above ground respiration and this below ground respiration. Because if you're just thinking about ecosystem respiration, those things get conflated. And so if you're really interested in thinking about what's happening to the autotrophs, what are the trees, what are the plants doing, how much biomass is accumulating, and at what rate, it's really kind of tricky because you've got this sort of confound of all of the soil heterotrophic respiration going on that can be this very variable contributor to how much carbon dioxide is coming out of that total system. And there are certainly ways that you can sort of try and back this out. You can sort of constrain it by looking at dark respiration at night of the system versus daytime respiration and parsing it across different levels of the eddy flux tower and all that sort of thing. But it's all kind of fuzzy. So if you really want to pull this apart and actually measure it, there are two ways to actually measure this soil component of this ecosystem respiration. And I'm going to highlight two for you. There are a number of other ones that people have come up with, but the two most common ones. The first one is a closed chamber method to estimate soil flux. And this is a diagram from Lightbore, which is an instrument company that does a lot of gas analyze, makes a lot of the gas analyzers that folks use. And basically the idea is you have a soil collar over your soil. You close the chamber over it, carbon dioxide builds up over time, and you have a continuous flow system that's measuring how that concentration increases over time. And then you open the chamber and it flushes and it's back to normal. Then you can close the chamber again and take another measurement. So when you close the chamber, that concentration starts going up, and the slope of this line is the flux, right? So you're closing it and you're waiting for it to build up, and then you're trying to take a very, very precise measurement of the slope of this line to estimate the flux back before the chamber closing disturbed this, right? So that's the closed chamber method. And then there's the method that Neon went with at all of their sites, which is the gradient method. So this is an image from Neon's documentation. And at each of these tower sites, you saw the picture of the tower, they also have 5 soil plots that are within the tower airshed. Because again, when they designed all this, they were thinking about, well, let's try and pull these things apart. So we want a way that will give us the ability to have continuous soil flux. And then we can subtract that out of the ecosystem flux. And now we have the ability to separate those two. And so the way that this works is you have continuous soil CO2 sensors at different depths deep in the soil. And by measuring the concentration precisely at the same time at each of those steps, you can then calculate a gradient and estimate what's coming out the top of the surface. So this is the diagram version. This is the real version that they have on their site to explain all the different sensors they have in each of these replicated soil plots. This particular picture is from Harvard Forest. But in addition to these soil CO2 sensors, which you have here, they have a deep soil moisture probe and temperature probe. They're taking ground level photosynthetically active radiation. They're taking temperature. They're doing a heat flux plate. They've got relative humidity, and all this data is being logged continuously. I mean, this is amazing if you're a data nerd. Like, you can get any of these individual things, or you can start putting them together to create these derived products, like what we were after with this question of soil flux. And this is what that looks like at Kanza, right? Very different from Harvard Forest, but the exact same sensors deployed in basically the exact same way, in the exact same layout. And this starts allowing you to do these type of larger scale comparative analyses that otherwise are almost impossible for individual ecologists or even small teams of ecologists to do alone, because it's just the infrastructure needs are so high. And they have people maintaining this year round at all of these different sites. And then just giving all the data away for free. So this is a sort of zoom in example here. So each of these are little PVC tubes that go into the soil to different depths, and then they drop down these probes. And down at the end of this is a Vaisala continuous soil CO2 sensor. And so they have 3 of these, and they're continuously streaming data back to this little hut, which is then on the network, which is continuously streaming data back to NEON headquarters, and they're doing all the processing and cleaning and everything else like this. So the idea behind setting everything up like this, spending untold amounts of money setting everything up like this, was that if you had all of this information, if you had the soil temperature at the different depths, you had the humidity at the different depths, if you CO2 at the different depths, then you could use an approach using fixed first law diffusion, which is a relatively straightforward equation, basically to estimate the flux out the top of the soil surface, which is what most ecologists care about, right? Because now you can start pulling apart what's going on with the net ecosystem exchange, right? So you see how the pieces are all fitting together. This was how NEON was designed. So what's the problem, right? What's like, we're setting up the tension here, right? This is a dramatic tension. Well, here's what happens. In 2015, I don't know, some of you know all the history here, but originally NEON was run by an organization called NEON Inc. And they won the first bid from NSF to implement the entire network. Around 2015, that all sort of crumbled because they were going over budget and there were some other issues. And so there was a transition from NEON Inc. to Patel, who is currently running NEON. It was a time of like much gnashing of teeth and consternation and all these sorts of things. But as a part of this reorganization, to make sure that they could finish building out the observatory on time and on budget, they had to de-scope a bunch of stuff. And they de-scoped a whole number of sites. Like there were actual additional sites. One of them that actually was quite controversial in the community is they got rid of the Baltimore LTR, which was going to be they were going to have an urban NEON site, which would have been very cool. But another thing that they did during this process was that they got rid of a bunch of planned data products. So NEON has this concept of levels of data. So level 1, level 2, level 3, and so on. Level 1 or even level 0 are like the raw data off the sensors, right? So like a level 0 even might be like the voltage off of the thermocouple or something like this. And then level 1 might be the temperature of the thermocouple, and onward and upward until you start getting these derived data products that most ecologists might be really interested in using. And again, the data product many ecologists might be interested in using in this case would be soil surface flux. And they had all the infrastructure in there to do it and a plan to produce a derived data product that would have done everything. But then they were hacked. And so they did all the hardware, they did all of this stuff, but then they just stopped. And they're collecting all the data and they're QCing all the data, and it's all super high quality, but it's all kind of raw. And what they said, their plan was that NSF Bio Directorate at NSF intends to engage the science community to develop the data products. So basically they were saying, we're gonna give people money in future grants to make up for the things that we couldn't do as part of setting up the observatory. And so this is where the actual science comes in. That was all the background setup. And this gets to that grant that I started with at the beginning. So I have interest in sort of microbial stuff and these sorts of processes. John Dobitz, my colleague, is really interested in sort of soil biochemical modeling and that sort of thing. And we both were sort of neon users from the early days, and we noticed when they descoped this, because we were like, we wanted to use that. And so we wrote this grant to basically fill that one little gap, of which there are so many still open, that are truly available for the community to sort of tackle and make a product of. So basically, our idea was, if you're getting all of the sensor information here from these buried sensors, like that's all good and that's all happening. Basically, we paired that with a field campaign where we used this closed chamber method to get an actual measurement in the field of the surface flux, and use that to validate and help parameterize this diffusion model that I mentioned earlier, to be able to hopefully start generalizing to this data product that then could be available for anybody at any time at any neon site. And so that was the idea. And basically, the field campaign, they happened over 2 different summers, 2022 and 2024, 6 different neon sites. The idea when we wrote the grant was that 3 would be close to me on the West Coast, and then 3 would be close to him in Minneapolis. He never actually came to the field work because he had some stuff going on. And so it was a very long drive for me, but we got to see a lot of really different type of ecosystems. And I should mention, we're both at primarily undergraduate institutions, and so this work was all being conducted primarily with undergraduate sort of summer research assistants. And just to give you a sense of what some of these sites look like, this is San Joaquin experimental range. This is in the Sierra foothills, it's super dry. Compare that to how green and lush Kanza was. This is in the Wind River experimental forest. This is in the southern bit of Washington State, right outside the Columbia River Gorge. So this is like old growth, like thick moss, like there was a foot of sphagnum. I mean, this was a totally different kind of ecosystem. This is in North Dakota at Chase Lake, which is wildlife refuge. Super thick prairie grass, but not tall grass prairie, actively grazed, really interesting site. This is in northeastern Wisconsin, almost on the border with the Upper Peninsula of Michigan. So this is again totally different kind of forest. And what we did is basically we got permission from Neon to basically go out to their soil plots and then put in soil collars right adjacent to it outside of the plot, so as not to disturb the continuous data, but near enough that we could assume many of the things were similar, and then used these light-4 infrared gas analyzers with this closed chamber method I mentioned to take measurements basically at the same place at the same time. So you could take the Neon data, you could use this diffusion modeling to figure out what the flux would be from that approach, and then you could go and you could actually measure it using this other method. And you could compare the two, and you could see like how well is this going to work, and how did that vary across different sites. And so what you get out of something like that, so we would spend about a week at each of these different field sites, in 2024, we actually used 2 different instruments simultaneously. So one of them was sort of robotically controlled and was 24/7, about every half an hour, taking a measurement. And the other one I actually had to, by hand, put onto the soil collar and take the measurement. And so that's what these 2 different color dots are. But you see, this is flux on the y-axis in micromoles of CO2 per meter squared per second. And then this is over time. And so you can see these really big swings, which in our case were primarily related to precipitation events that happened while we were there. So if you get a precipitation event, you get a big spike of soil respiration as that re-wets. And then you see that sort of settle back down. And then depending on the temperature in the day too, you might see a really strong diurnal swing, or it might be kind of just like chilled out for the day. So taking all of this data, basically that was the sort of field truthing. And then John's focus was on developing an R package that basically handles the neon side. So basically, from whichever neon site at whatever time you want, it acquires the data through the neon utilities package, which Claire is the author of. Does some gap filling, deals with QA flags, does some interpolation to handle slightly different depths. And then using these model approaches, computes the fluxes across layers, and then spits out that predicted soil flux, the uncertainty around it, and any sort of quality assurance flags, just to let the user know what's going on. And I should mention that this, the R package that does this has been on CRAN for, I don't know, 7 or 8 months now. It's called Neon Soil Flux. Anybody can download it, use it. It works now, and we're making it better as we go. So just to give you a little sense diagrammatically about how this works. So basically, this over here on the left is how these probes work at the neon site. So you can think of each of these colored dots as the location of a sensor in a soil depth of the soil profile, right? So this blue here is soil water content. So you might be measuring soil water content at these 3 different depths, but maybe one of the sensors at a certain depth died at that neon site. You gotta handle that. And maybe the depth is not exactly the same as where the temperature is in the adjacent sensor, right? Which might be a slightly different depth, and maybe the bottom 2 for that one didn't work, right? You have these differences that come up because this is like actual field ecology, and things break in the field. And you also have those soil CO2 sensors. So the first step is to sort of harmonize those by basically interpolating and calculating them so that they're all the exact same depth. And then you can use that set of measurements that are now sort of harmonized and all of the appropriate set of co-located depths at this site, use that to calculate the gradients, estimate the diffusivity, and then calculate the flux. And there are a lot of different ways that you can do this, and this was the thing that we learned the sort of deeper we got into it. And the real core challenge here, so this is, we have a paper that we're writing on this in Methods in Ecology and Evolution, and we're just about done with the revisions to send it back. But this is just a little excerpt, and this equation here, this is Fick's law, right? So it's basically saying you're looking at the derivative of carbon over depth, and then that's multiplied by this diffusivity constant, right? So this dA is this diffusivity. This is the hardest part of this entire thing, is figuring out what this is. And this is the reason this was descoped, right? Because if you could just set this to a flat number, like the neon folks could have done this, like they could have done it. The problem is doing this right to make a data product, like fully properly, would have required calculating this very specifically across every single different neon site from the field, doing the actual measurements of diffusivity directly next to those soil plots. And that would have been hugely time consuming and hugely expensive. So I totally understand why they cut that out, because they were getting all the raw data and then somebody could sort of help get this across the finish line. An important take home from our analysis here is that, as expected, getting this diffusivity parameter right can be challenging across these diverse sites. And this is something we're actively working on sort of untangling now. So this is just an example. So here on the y-axis, we have this diffusivity parameter. And each of these 6 different facets here is one of those 6 different field sites. And on the far left of each panel, we have the field estimated diffusivity. So here, if you know the truth that's coming out the top, you can back calculate the diffusivity with some pretty good accuracy, because you know what's coming out the top, and you know everything else. Basically, we have this additional constraint, and so you don't have to sort of fudge or estimate what that diffusivity is, because you can constrain the system and figure it out based on those soil surface measurements at that time in that place. And so that's the far left. And then these 2 adjacent to it are 2 alternative ways of estimating that diffusivity parameter. One by Marshall from 1959, and one from called Millington Quirk, which is from 1971. That are sort of standards in the field. And what you can see is that at some of the sites, one of the 2 will be spot on with what we measured in the field. right? So this is just a way of estimating this parameter, not knowing exactly what the truth is. And so sometimes it'll be one parameter and it'll be like really close. And then at a different site, it'll be the other parameter, but it'll be really close. But then sometimes, just kind of hiding over here, both parameters won't work. And so what's been really kind of exciting for us, frankly, I mean, this is the challenging part, but it's also the exciting part, is trying to think about why. And we've had some really interesting discussions thinking about why the parameters of particular sites are causing the model to behave the way it does. And just as one example here, Santa Rita Experimental Range is south of Tucson. When we went there and measured, it was 115 degrees in the shade. It's like sandy desert, and the sand surface was like 55 Celsius, like really exceedingly hot. And it's this very, very mineral sandy soil, exceedingly dry. And what you get is you get this heat bank effect, where in the soil surface, right, at night gets cold quite quick because it gets cold in the desert at night. But under lower in the soil profile, it's like a heat battery, right? It's like a sand heat battery, so it stays really warm. And in the day, the soil surface heats up really fast because it's close and the sun is really intense and everything like this. But the soil subsurface, because there isn't a lot of water to transfer the heat, heats up much more slowly. And so you get these really interesting temperature patterns that you don't get in a normal sort of organic soil that's more moist. And so you start seeing these patterns and you can start pulling them out. And that's kind of what we're working on now, is thinking about when is it that one of these sort of off-the-shelf estimates might be really great at a particular site or set of sites. And again, we're trying to apply this across the continent at all the keynote sites. So thinking about what things you can generalize and what places you might need to think carefully has been really interesting. This is super busy, but I just want to give you a sense of how we're thinking about this. So I'll set this up for you, the colors first. So the orange dots here, these are field measured flux values, right? So this is the sort of closest thing that we have to sort of empirically measured truth. Then we have these two models, Marshall and Millington Quirk, the little purple line and the little green line, and both of those have error bars. And each of these is over 4 to 6 days, roughly speaking. And going from left to right, these are the 6 different sites. And then the different rows of all of these different panels correspond to these different approaches to calculating flux. Because sometimes you want to use just a sort of linear extrapolation of your 3 different soil sensors. But sometimes you want to just use the bottom 2 and not the top one for some reason. And so you look at the gradient between the 2 lower soil sensors. Or maybe you look at the top soil sensor and the lowest one and skip the middle. Or just the top 2 soil sensors, right? So you can break all of this apart, depending again on some of these sites for specific parameters. And that's what we're doing here in each of these different rows. And what I want you to take home from this is that in some cases, those differences matter a lot. And based on the conversations John and I have had about this, they also tell you a lot about the soil dynamics of what's going on in that site. So in some cases, this measured line basically falls right on top of what they're both models, both of these other constant estimation strategies are predicting. Sometimes it falls right between them, like in this case here, in this case here. In this case here, it's right between these 2 extremes. And in some cases, there's something different going on, right? That is not captured by these models that have been developed and used for many decades by the soil science community to estimate these sorts of things. And those sorts of sites are going to need a little bit more insight, a little bit more planning to think about. But it was really, I think, from a like field ecologist background, going out there and being in those sites and then seeing this in the data, I was like, oh yeah, like that makes sense. Like if you know, when I was digging the soil pit down there, like, yeah, this was like a lot cooler and this was a lot warmer. And like, here's what's going on in that type of site versus this other one. And then it shows up in the data like this and influences flux predictions. So all of that is to say, like, you can get really lost in the weeds in this stuff, But if you back it out, so this is now not just the week scale, but this is using those various parameters to basically calculate soil flux over an entire year at the 6 different sites. And now the orange dots are the points that we measured, right? And what you can see is that when you're looking at across the scale of sites that vary across, what we see at these 6 sites across the continent, the estimation in general is actually pretty decent at getting in the right ballpark, even without doing the really intensive, expensive ground truthing in every single different neon site that you would need to make this perfectly precise. And so what we're building into the package now is sort of all of these different options for the user, and also some general frameworks of like, if it spits out a number that looks different like this, have you considered that maybe the site needs adjustment in this way or something like that? And so we're trying to make it a little bit more user friendly for the user to sort of parse through the whole set of options that we pulled out of literature and put into this package. Okay. So that's just sort of where I wanted to sort of stop on talking about that research project. It's still in progress, but it is coming to a close, and John's sort of carrying it forward in this forecasting prediction framework. So I wanted to talk a little bit. The organizer said I could indulge myself a little bit, and I'm also super excited about macrosystems data and macrosystems thinking in teaching. And so I wanted to share a couple examples, a few related to a faculty mentoring network that we did as part of this grant, and then some that I've just used in my classroom for the last 8 or 9 years. So one thing you may not have noticed, if you didn't have a real careful eye there, at the bottom of all these Picaros in the neon shed, this is a Lightcore 850. Lightcore 850 used to be called Gas Hound, for any folks who have worked with soil flux instrumentation. But it's an infrared gas analyzer that measures concentration of water vapor and CO2, basically as a little pump. And it just uses infrared absorption of the gas of these particular gases and tells you what the concentration is. They're great. Those Lightcores are exceedingly expensive sort of precision instruments. These are not cheap, but maybe like $5,000 or $6,000 each, and still really high quality instrumentation. So as part of this grant, we included funds to basically run a faculty mentoring network. We called it the Neon Soils Macrosystems Faculty Cohort. And basically we bought all the participants one of these Lightcore 850s. And the idea was to have each of them learn how to use this piece of instrumentation, telling them about its context with how Neon uses instrumentation like this to do their field measurements and estimates of gas flux. And then both sort of allowing them to demonstrate that to their students, like in person, like with their hands in a class. One thing I will often do in my ecology class is just have that on and running and close all the doors in the classroom. I don't know if any of you have ever done this who do gas exchange stuff. And at the end of an 1.5 hour class, carbon dioxide is a lot higher in that room. And you just start doing some really fun sort of back of envelope calculations with the students. Like, how many breaths did you take? How big are your lungs? Let's do some quick calculations. And they're like, oh, wow, like the carbon dioxide tripled or quadrupled in an hour. And it just makes things a lot more tangible. The other thing that I was demonstrating to people, this is an example from a module I taught in one of John's classes at Augsburg, actually, it was a calculus class. And I brought all these soil microcosm jars in with these septa, and you can basically take a syringe and then you can inject into the flow of this 850 to get a spike measurement of the carbon dioxide in the headspace. And so you can basically see how much soil respiration is happening in these different jars. And so the idea here is this was a math class. This was not necessarily like a field ecology or biology class. But the idea was to like give students some real tangible experience using something they learned in the class. In this case, it was calculating Riemann sums, right? So this is a way to do definite intervals. So you can do left hand, you can do right hand, you can use trapezoid methods. Basically, you're calculating the area under these curves. And then you're using that to back out the concentration of carbon dioxide in the headspace. And so this was not using any R or any programming at all. They had some Excel and they had some measurements and they put it together. This is what one of those spikes would look like when you inject into that stream, so you get this spike. And then you use the formulas they've been learning in class to calculate the area under the curve, basically. And you do that with some unknowns, you do that with some known standards, and then they can practice using some of their linear regression models and interpolation to calculate what those values would be for those unknown samples. So it's like very hands-on and also directly relevant to what they were doing with calculus. So I thought that was kind of fun. And so I put together for this faculty mentoring network, I put together a video sort of for these instruments, like, here's how you set it up, here's how you install the software, here's how you set up the little background injection thing and the septic port and the microcosm chamber, here's how you inject it in, here's how you take that data and put it in R and all that sort of thing. And then we had participants go and run with it in whatever class they were teaching that semester. So these are just a couple examples. This is an example from a community college professor, actually in Johnson County in Kansas. And on their campus, they have a sort of restored native prairie, sort of little patch that they have on campus. And she with her class went out and they dug at different soil depths, soil from near the surface, in the sort of lawn, and then in the native prairie, and then at a deeper depth in the lawn and the prairie, and then they incubated it in the jars, and then they measured how much carbon dioxide was coming off in the headspace. And they found super big differences, which was really exciting for them and for the class. And then this is another one. This is an example. This was a fellow at Gonzaga University, and he developed a whole thing for his class, building off the teabag experiment. Some of you may be familiar with the teabag decomp experiment, but he basically did the teabag decomp experiment in these little juice jars, and students could add different levels of water, or they could change the acidity of the water that was added, or they could change the temperature that the bottles were incubated in, or whatever else. And then they measured the carbon dioxide in the headspace over time, and they could start thinking about the principles that were going on during decomposition using this instrument and this framework. And then this ended up getting published in Course Source because he was very organized. Okay, so that's sort of the end of talking about what happened with that grant. And this last one, this is sort of more my pet stuff. This is teaching data analysis in classes where you don't necessarily have time to go to the field. You don't necessarily have time to even have a lab in class or something like this, but you really want students to be actually doing science. Like you want them to get that discovery feeling, which is kind of hard in a lecture course sometimes. And I think, and I have been telling anybody who listened for many years that I think Neon is absolutely the best way to do that. I think that what is provided by Neon in terms of its data sets make this tractable, even dare I say scalable, for students to do independent original analysis that is self-directed using coding and quantitative analysis. And the reason it's possible is because the Neon data are all the same but different. And there's very little other data out there that is as the same but different. And what I mean by that is the formatting of all of this stuff at every site is identical, but the data is totally different, right? So the spreadsheets are set up exactly the same way. The row names are the same. The column names are the same. Like you download them with the same package. You like, all of this is exactly the same. All the documentation is the same, but the data is all different. And there are so many different sites that students can pick whatever they're interested in. And they have that autonomy. But you, as the instructor, have the structure of the Neon data to fall back on. And so you don't have to solve 50 different data problems from 50 different students, which will kill any attempt to do this in any kind of class. And some of you may have experienced this, yes. And so this is just an example. This is the Neon data portal. And if you have not poked around on here, I hope all of you have, but it can be a little bit overwhelming, but it's also really, really powerful. And you can really dig in here and find things that you personally or your students can get really excited about. Like maybe they don't care about soil flux, and it's not tangible, but they are really into beetles. Like they love beetles. There's a cool beetle data set. Or maybe they like birds. There's a great bird data set, right? Maybe they're into mammals. Maybe they're into pathogens. A lot of my students are pre-med or pre-health. they love the packaging data sets, right? Thinking about whether there's Lyme disease in the ticks, or whether that's in the rodents and how that's spreading, or what's in the mosquitoes. Like, do they have what kind of mosquitoes and where are they and how is that spreading across the US over time? And they get really excited about those because they have public health implications. So again, you have all of these different sites, but they all have the same data. Like, this is incredible. Like, LTER is fabulous, but it's very different. Every LTER site has a different set of data done in a different way. It's not standardized. It's not programmatic in the way that Neon is. And the end point of a project like this, of a class like this, is a final report that the students write. So this is a zoomed out view of a Word doc of an undergraduate student's report. So we have, 5 or 6 pages of prose text where they're writing up their analysis and thinking about their questions and saying what they did. And then they're making some figures, right? And then they're citing some sources. Like, they're doing the whole scientific thing on data that maybe nobody has ever looked at before. And that's possible because Neon cleans it and Neon standardizes it. That would not be possible for a novice otherwise. And so the basic framework here that I have come to over many years doing this many different ways is relatively straightforward. Basically, you have your Neon data in the cloud. Students can browse around on the web browser. They can find the data sets that are interesting to them. There's a script that I provide to them that I'll show you in a second. It basically downloads whatever they want for their particular site at their particular time to a text CSV file of that data. Right? Step 1. Step 2 is they write their reports in what's called an R Markdown or a Quarto file, which is basically a literate programming approach to put together prose and code in the same document. It also, doing it this way, enforces reproducibility, because you cannot either knit or render one of these documents if all the pieces aren't in place. And so as an instructor, if you're trying to troubleshoot some student's thing and their directory is chaos, right? It can be exceedingly difficult, if not impossible. But by saying your thing needs to be able to knit, start to finish with no errors, you're enforcing that all of that works and all the things are in the right place. So it makes helping them a lot easier. And then finally, it produces this lovely report that they can then share. So for those of you not familiar with R Markdown or Quarto, this is just a quick example from the Quarto website. Quarto is basically the new version of R Markdown, but they're very similar conceptually. You have some text, document metadata at the top, a title, a name, a date, something like this. You have some text and prose, which gets rendered into section headers and prose text. And then you have what are called code chunks, which are basically code, in this case in R, but this also works with other programming languages if you're more comfortable. And then when this gets rendered, it turns into this lovely document that you wouldn't necessarily know came from this code thing. So they could, like, I have students who learn this in my class, and then they start using it in all their other classes. Their instructors that they're using it in don't know any R, don't want any of this, right? But they're just giving them a Word document or an HTML document or a PDF. And it's just nicer and crisper, and everything is rendered. So students really get into it. This is that script, that download script I mentioned to you. I provide this for them as a template, and all they have to do is change these parameters, right? So this is a function in Neon Utilities called load by product. And basically, this DP ID is the data product ID. This site is the site, the Neon site. And then you're saying, I want to check if it's super huge. We don't want to download it to a student laptop. You pull out whatever particular data set you're interested in. So this student was interested in, for example, macroalgae biomass. They were looking at stream data. And then you write the CSV. So very scaffolded. But the students have a ton of freedom, right? So first thing is data product ID. They can just browse on this website and find the thing that they think is cool, right? And they don't have to know any coding to do that. They just go to the website and they're like, birds are awesome, bugs are awesome. Some microbes, there are a few, think microbes are awesome, right? Or whatever it is. And they get this data product ID right from the website. They drop it into the script. And then they pick a site, right? So this TECR, they can just go to the Neon website and browse on the map. Students pick things that are closer to their hometown. They pick someplace they've always wanted to visit. They pick it because it's called Disney, and they've always wanted to go to Disneyland, right? Like, students pick these things for the weirdest reasons, but then they get so attached to it. And it does magic for getting them through the difficulty of learning this kind of stuff. So they didn't take that code. They can read about the site. It has great information and pictures and everything else on their websites. The students can really kind of get a feel for it. And then they just drop that into the script. And that downloads the data for them. And then they're done. They don't need to use the web anymore. They don't need to use the portal anymore. And then all they're doing in the R Markdown, it's really straightforward. They're loading a couple packages. They're reading that CSV in, and then they're just making some plots, and then they're writing about it, right? So it's not. It can be as complex as you want it to be, but it can also be very simple. The students who come into my classes are terrified of p-values. They ostensibly have taken biostats, but they've probably forgotten it because it was 3 years ago. And they generally don't have any programming experience and are fearful of dealing with computers in this way. But when they do it, they get so excited because they all can do it. Well, it's normal. But they get so excited. It's really amazing. And then they're creating these beautiful, technical, polished reports that work really well. So this is just one example. I'm on the last couple slides of something rendered. This is the type of thing a student might come through, and they might say this particular species seems to have more dry mass at this site. And then they do a little natural history research on this species, and they think about why is this species the one in that site that is really common? Like, what is this? And then they can tell a story about it. They can find some pictures online. They can start thinking about the connections of those narratives. And then finally, that all sort of gets put together in this rendered report. And the sort of big picture here, right, is like the goal, I think, to get students excited about science. And like, I really feel like I missed the boat as an undergrad getting jazzed about science in my classes. Like a lot of that happened in like summer internships or something like this, right? And then certainly in grad school where you had your own projects. But like, if you could give undergrads a taste of that, like that's transformative. And not all of them are going to be able to do REUs. Not all of them can afford to work in a lab because maybe they're working 3 jobs or they're taking care of their little cousin or something, right? But if you can give that to them in a class that's authentic but bite-sized and well scaffolded, that's really, really powerful. So like Neon lets that happen. Like, I haven't found a way to do it without Neon. The self-motivation that comes from ownership gets them over this high activation energy of being kind of intimidated by this stuff. And then finally, tying back to this macro systems theme, in the last week of classes, they all present on what they've done. And so now we have a class of 20-something students, each of which is presenting on a totally different Neon site, on different data. But then this sort of magic happens at the end where they're now start seeing things at much bigger scales, right? They're seeing how different sites vary and how those are located in relation to one another and how those change over time. All these sort of bigger things. Well, they were deep in the weeds of their particular site, their particular analysis they didn't see. But then we have these really great discussions at the end, sort of bringing it back out to that big picture, which works really well. And I have a little PS here. LLMs and generative AI have really kind of thrown a wrench in some of this, and I'd be happy to chat about that. I have lots of thoughts and some strategies, but I mean, good things and bad things, for sure. So, in conclusion, the big take home here, macrosystems research and cheat chain works really well with Neon data. I really, I think the thing that I tell everybody who will listen that I think a lot of ecologists miss is the thing that I think is transformative about Neon is not its scale, it's not all the different measurements it's taking, it's how it's handling its data. The fact that they are standardizing it the way they are, the fact that they're allowing those data sets to be merged, the fact that this is all open and available right away, programmatically, with the package in multiple different programming languages, like changes the game for getting students working with real data. Because you could do all this stuff with some contrived CSV that you came up with, and everybody in the class does the same CSV, and it's fine, right? You can do that. That's practical. But it's not authentic because you kind of know what's in there, and everybody's going to kind of be doing the same thing. But with Neon data, they can all be doing original analysis in a place that you would have no idea what they're going to find, and so it's kind of exciting sometimes. You're like, "Whoa, that graph's really cool. What does that mean?" And you can have that moment together that most people don't get to. That's me, right? So that's really cool. So kudos to Neon for that. And then, yeah, I'll just end it here. Happy to take any questions. That's my new email, which is kind of weird. After 9 years of stuff. And this is the Santa Rita Tower. Outside, which is, so thank you.
All right. I'll get the mic for 1 second.